In the previous video, we introduced RNA sequencing as a method that sequences fragments of RNA instead of DNA to perform transcriptome profiling. In many cases, you will need to use a different approach to mapping RNA sequencing data than you would for DNA sequencing data. This video will discuss the different options for mapping RNA-seq data back to the reference genome and will introduce the splice-aware aligner known as TopHat, explaining what it is, why we need to use it, as well as how it works. Splicing is a method of RNA editing that removes non-coding intronic sequences from pre-messenger RNA. When performing RNA sequencing, we are predominantly sequencing the mature forms of RNA, meaning that the mRNAs that we sequence will be spliced. The amount of splicing that occurs in your model organism will determine what method you use to map your RNA sequencing data. For organisms with little to no splicing, such as E. coli and yeast, you should map your reads using standard aligners such as Bowtie or BWA. For organisms with extensive splicing, such as mice and humans, you will need to use a splice-aware aligner, such as TopHat. One exception to note is that if you are particularly interested in splicing in an organism with very little splicing, you should then also use a splice-aware aligner. Another exception to note is that if you do not have a reliable genome assembly or set of gene annotations, then an alternative approach is to first perform de novo transcript assembly and then map your reads to the transcriptome and not the genome. How does splicing affect read mapping? RNA splicing creates sequences of RNA and thus cDNA that do not exist in the reference genome. When we fragment our RNA to make a sequencing library, any fragments that span a splice junction will not map to the reference genome using traditional read aligners. Instead, we must use tools that take splicing into consideration, such as the program known as TopHat. TopHat was created by Cole Trapnell, who also helped to develop the Bowtie read aligner. Currently, TopHat is a collaboration between Trapnell and his group and the Center for Computational Biology at Johns Hopkins University. TopHat was designed to perform de novo splice site discovery. There are several versions of TopHat, and as with Bowtie, we will focus our discussion on TopHat 2 instead of the original TopHat program. TopHat 2 uses two sources of evidence to identify splice junctions, thereby allowing reads to be mapped to these particular regions. The first source is to stitch together segments of reads that are mapped independently, and the second is to pair together coverage islands from continuously mapped reads. I will explain in more depth how these methods work, but first, I want to give you a general overview of how TopHat 2 operates. The first phase of TopHat is to use Bowtie to map reads to the transcriptome. This works by creating a reference index from all possible known RNA sequences in your model organism. This step is optional, but will greatly speed up your TopHat run. To do this step, you must, of course, have a gene annotation file for your reference genome. The next phase of TopHat is to then map your reads, or any unmapped reads from the previous step, to the reference genome, again using Bowtie. This step will include read segment mapping. Using the genomic alignment results in this step, TopHat will then generate a library of potential splice junctions and will take all remaining unmapped and poorly mapped reads from the previous steps and map them to an index of splice junction sequences. TopHat was originally designed to identify splice junctions via what it calls coverage islands. Coverage islands are first identified during read alignment to the reference genome. During this step, what we often see are islands where reads are able to map along the genome. TopHat makes the assumption that these reads do not span splice junctions, but instead correspond to purely exonic sequences. TopHat then assembles these continuous regions into what it refers to as pseudo-consensus sequences. These sequences are then extended on both ends to compensate for loss of read coverage around splice sites, and potential splice site acceptor and donor sequences are identified, and islands are stitched together to build a library of putative splice junctions. Any remaining unmapped reads are then mapped to an index built from a 2KB sequence centered around the splice site junctions. New to TopHat 2 was the incorporation of read segment mapping. My understanding is that TopHat 2 relies more heavily on read segment mapping for splice site discovery than it does for coverage islands. The way that this works is that TopHat 2 takes reads of 45 base pairs or longer and breaks them into segments, and then maps these segments to the genome independent of one another. When segments map to different locations that are within some distance of one another, TopHat makes the assumption that these segments are mapping to distinct exons. Segments from the same read are then stitched together using nearby splice site acceptor and donor sequences to generate a library of potential splice junction sequences. And as we saw with coverage islands, unmapped or poorly mapped reads are then mapped to these potential splice site regions. The incorporation of read segment mapping into TopHat 2 has improved its ability to perform indel discovery and also allows for the detection of gene fusion products. TopHat 2 has a number of additional features that make it a flexible and powerful tool for RNA-seq read mapping. For instance, TopHat 2 has additional algorithms that improve indel identification in spliced read alignments, and it can distinguish true spliced reads from reads that originate from processed pseudogenes that have been transposed into the genome. TopHat also contains algorithms that allow for the identification of gene fusion products, and will also take in a set of known genomic variants to improve your RNA-seq read mapping. One last thing that I want to note is that as of February 2016, TopHat 2 has entered a low maintenance and low support stage, and is likely to become deprecated down the line. Instead, Trapnell and the Center for Computational Biology at Johns Hopkins University have been developing a general-purpose aligner called HiSat2 that they claim will supersede TopHat as a more accurate and efficient tool for spliced read alignment. Now, before you run off to use HiSat2, know that it is still in beta testing, and TopHat 2 will of course still serve as an excellent tool for spliced read alignment. But I wanted to bring this to your attention so that you may incorporate HiSat2 into your analysis pipelines in the future.